Hello, everyone. Welcome to Lunch Break Science and Happy New Year. I'm Ariel Johnson, your host from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries. Today, we have a wonderful episode focused on primate sensory perception. Before we jump in, though, we'd like to thank the Ann and Gordon Getty Foundation and Camilla and George Smith, whose generous support made today's episode possible. Now, let's bring on today's guest, Leakey Foundation grantee and Science Executive Committee member, Brenda Bradley. Brenda, thank you so much for being here with us.
Thank you for having me.
Brenda's joining us from Washington, D.C., where she is Associate Professor of Anthropology at the Center for the Advanced Study of Human Paleobiology at George Washington University. Her work takes a multidisciplinary approach, bridging primate behavioral ecology and evolutionary genomics. Her collaborators range from primatologists studying lemurs in Madagascar to chimpanzees in Uganda and beyond, so there were too many locations to add to our little map there, but she'll be talking about some of them later. Before we hear more about primate senses from Brenda, if you are watching this episode live, please post comments or questions for us in the chat, and Brenda will answer them live during the episode. The earlier you get those questions in, the more likely your question will be featured. So, Brenda, my first question for you is, when contemplating our evolution, why are we interested in looking at the genetics of primates other than us humans?
So my lab is particularly interested in understanding how can small genetic changes result in such huge differences in how animals look and behave and function. Basically, we're interested in the genetic basis of diversity. And if you want to understand diversity, there's no better group to look at than primates. If you think about primates, particularly compared to other mammalian orders like rodents or rabbits or ungulates, primates are arguably the most diverse of these groups. And that's true for almost any phenotypic realm you can think of. So consider body size. Primates range in body size from a tiny little mouse lemur that will fit in the palm of your hand up to a huge silverback gorilla. So a huge range of variation in body size. Primates also differ a lot in the types of habitats they live in. So primates are found in everything from dry grassland savannas to tropical forests. And then even within a forest, primates will be found in different microhabitats, some living in the canopy, some living on the forest floor. So there's a lot of variation in habitat. Primates show a lot of variation in social structure. Almost every possible configuration of a social system that you can imagine, you see it in primates. Everything from monogamous pairs to polyandrous groups to huge multi-male, multi-female groups with hundreds of individuals. So primates also have a lot of variation in their sociality. And I could go on and on with examples that primates show variation in diet and variation in activity cycle. And of course, primates are extremely colorful. Primates are almost certainly the most colorful mammalian order, showing a lot of variation in aspects of pigmentation. And this is a topic that my research group is particularly interested in. So ultimately, we're trying to understand what is the genetic basis of the origin and the maintenance of all of this phenotypic variation. And we go about this by looking at a lot of different individual case studies.
So during your talk, you'll be focused more on the variations in primate sensory perception. But as you mentioned, your lab, the Primate Genomics Lab, looks at a much broader range of primate variations. Could you tell us more about your lab and the different projects you're working on? And then we're also going to share a link to the Primate Genomics Lab in the chat.
Great. Yeah, so we're a genomics lab, but I think we see ourselves as more of a primatology group. So for us, genetics is a way to understand aspects of primate ecology and evolution that we might not be able to get through observation alone. But pretty much everyone who works in my lab, in addition to doing genetic bench work, they also do field work or museum work. It's important for us that we're never studying the genes as simple molecules, but always thinking about our broader questions, our broader goals are to better understand the ecology and the evolution of the primates. And we go about this in several ways. So my earlier research was largely looking at gorillas. so population genetics and mating structures of wild gorillas. And we're still really interested in these types of questions. We have projects now that are looking at similar things, but particularly in lemurs. So we have projects looking at conservation genetics and population genetics and mating structures and sexual selection in lemur populations. So we're still doing a lot of those types of kind of traditional population genetics projects for both apes and lemurs. But then there is another set of projects in my lab looking at genes that might provide clues to the evolution of specific traits. And some traits that we're interested in are things like hair growth and hair texture. And we have this ongoing project looking at the evolution of pelage in primates. This is largely in collaboration with Jason Kamilar. And we're looking both at the genetic basis of hair variation and then also collecting detailed phenotypic data on hair. So we go to natural history museums and using their research skins, collect a lot of phenotypic measurements of hair variation. And then we're just broadly interested, not just hair, but in the broad sense, we're interested in ornamentation and coloration and differences like that in primates. So here you see a couple of species of lemur where you see the male and female of these two different species. So you see variation between the species, including in their eye color, and then variation between the sexes within the species.
That's so beautiful, that image.
And then we're also interested in genes that might underlie variation in aspects of communication or behavioral style. So genes like FOXP2, or we also have some projects looking at variation in serotonin receptors. So we work on a wide range of topics, everything from hair to language.
It's got to be really fascinating to have that kind of breadth to work on. I mean, you really kind of get that bigger picture.
Yeah, it's been great.
Your lab also took on an additional role, great relevance to the world today. Tell us about that.
Yeah, so, one of the standard procedures in my lab is PCR. Almost every project that we do, whether it's language or pigmentation or color vision, pretty much every project starts with extracting DNA and RNA and then doing PCR. And now everybody knows what PCR is because of the PCR COVID testing. So it became apparent to us early on that all of the protocols that are used in PCR-based COVID tests are the same protocols that we use in my lab. So we collaborated with the GW School of Public Health. This was largely spearheaded by them in connection with a private company, Curative. And over the summer of 2020, my lab got transformed. We did this in about a week, completely revamped the lab. There you see a whole panel of PCR workstations where basically my lab then was participating in this PCR-based COVID testing. And one part of that that was particularly interesting to me is that several of my students who had been up until that point working in the lab on projects about things like primate population genetics, they continued to work on the lab doing the COVID testing. And I think it was interesting for them to see that, oh, this set of skills, this set of lab skills that they had acquired working on projects about primate evolution were skills that they could directly apply to something that was more high stakes in the world of public health.
That is so amazing that you saw that opportunity and that your students got to get this great experience.
Yeah, I think it was really great for our lab to have that opportunity to participate.
So when did you first become interested in studying human evolution?
I grew up in a really small town in rural prairie land, Colorado, and I had a great high school science teacher, but I wasn't really exposed. Yeah, here's a picture from my town of the sports field, which is kind of the center point. But I wasn't really exposed to evolutionary biology and definitely not anthropology until I got to college. And the real turning point for me there was when I met Leanne Nash in my junior year. And Leanne at that time was studying, she had a captive colony of galagos. There at Arizona State, there's Leanne. And I had an opportunity as an undergrad to help out on her research project with the galagos. And that was just a game changer for me. It was my first time participating in actual research, and I was hooked. So this was my junior year, I immediately changed my major from bioengineering to anthropology. And I was particularly fascinated with primates. So Leanne also hooked me up with a project at the Phoenix Zoo. This was a project studying Hamadryas baboons. And Hamadryas baboons, yeah, here you can see them. And the thing that fascinated me most about these Hamadryas baboons are their crimped hairstyles. They have these really amazing hairstyles. And so it's interesting to me that now, more than 25 years later, since I first was working on that project, I'm back to studying, these aspects of hair variation in primates, but now from a genetic level.
It is, you never know where that, inspiration will come from.
Or come back from years later.
Or come back from, yeah. So what was your first experience out in the field collecting samples like?
Yeah, my initial dissertation plan was to do population genetics of wild putty-nosed monkeys at Mondika Research Center. So this was a site that was established by my advisor, Diane Doran Sheehy. And I thought, okay, my dissertation is going to be to go out to watch these guenons, to follow them. When they defecate, I'll collect their DNA samples, and then I'll use those DNA samples to study things like their mating system, dispersal patterns, migration, basic genetic variation. And I quickly realized that I was not suitable for that type of field work. I really struggled. I struggled on most days to find the monkeys. And then if I could find the monkeys, then I struggled to keep up with them. And then if I had the miraculous occasion to actually see a monkey defecate, because that's what I was there for, to get the DNA samples, then often it would fall from the canopy and then I just couldn't find it on the forest floor. So I was getting increasingly frustrated. And at that time, Diane Doran Sheehy, my advisor, was there at Mondika. She was in the process of habituating the wild gorillas there. And she said, why don't you just take a break from the monkeys for a day and come out with me and follow gorillas? So on that day that we followed gorillas, when you follow gorillas, you find feces everywhere and plenty of it. And I found myself thinking, oh, I'm working so hard to find a single fecal sample of a guenon monkey, and yet there are gorilla feces everywhere. Why am I not studying gorillas? And then it's like a light bulb went off, and I'm like, yeah, why am I not studying gorillas? All of these same questions that I have about the guenons, about the mating structure, the migration patterns, the dispersal, the genetic variation, the relationship between social groups, all of those same questions are also applicable to gorillas. So basically on the spot, I switched from guenons to gorillas. And then over the course of my time in the field, I was able to collect thousands of fecal samples and hair samples from the wild gorillas at Mondika. So it was one of those seemingly obvious things that just didn't strike me until it was just one quirk of one day in the field where I thought, oh, there's a way that I can quickly change this project to make it much more feasible and much more suitable for my particular field skills.
Well, you have to be adaptable out in the field, and you've come a ways from that first field time, and now you're developing new techniques and new ways of applying techniques in the field. So tell us about that part of your research, or yeah.
As I mentioned before, so we're a genomics lab, but almost everyone in my lab also does field work. And even if you're just studying the genes, it's still so important to understand the natural environment and biology of the animals. And in fact, we're trying to develop ways to do a lot of this preliminary lab work to do it in the field. So with my former student, Elaine Guevara, we've developed a sort of lab in a backpack that we've tested at, this is a photo from Beza Mahafali Research Center in Madagascar. And it's basically a little backpack that has pipettes, the basic reagents that you need. It's a mini PCR machine, a mini gel rig, and you can do a lot of kind of basic genetic tests at remote field sites. If you have some solar panels, and you can bring in this backpack, and of course, you're not going to be able to do full genome sequencing in the field, but you can do very simple genetic tests like like doing a test for color vision, as I'll talk about in my presentation. Those are the types of tests that you can do even at a remote field site.
I'm just really excited to hear your talk, and I wish we could like hear a talk on all of the different areas of research you go into. But before we turn the virtual floor over to you, Brenda, if you are enjoying this episode of Lunch Break Science, be sure to subscribe to our YouTube channel or follow us on Facebook or Twitter to receive reminders of upcoming episodes. And if you hit the little if you're watching on Facebook, if you hit the little bell, you will be sure to get those notifications. So now let's turn the virtual floor over to Brenda and hear more about the variation of primate sensory perception.
All right. So as I said, fundamentally what my lab is interested in is understanding how genetic variation can generate phenotypic variation, and selection can then act on that in shaping adaptation and diversity. And I would argue that color vision is a particularly great system for studying these types of connections, especially in wild primate populations. It's also a really interesting system if you're studying primate evolution, because full trichromatic color vision is unique to primates. Most mammals are red-green colorblind. So before I get into the actual research, let me just remind you and clarify some of my basic terminology. So when I talk about full trichromatic color vision, I'm referring to the type of color vision that most of us have, the ability to distinguish between reds and greens. And that's in contrast to being red-green colorblind. And I will note that there are different types of red-green colorblindness. Now, you probably remember from, I don't know, 6th grade biology class that your color vision comes from the photoreceptor cells, or the cones, in your retina. And those cones in your retina contain photopigments, or opsin proteins, that are sensitive to different wavelengths of light. And in order to have full trichromatic color vision, as most of us do, you need to have 3 different types of opsin proteins in your retina. So you need to have opsin proteins that are sensitive to short wavelength light, some that are sensitive to medium wavelength light, and some that are sensitive to long wavelength light. Now, the gene that codes for the short wavelength opsin protein, it doesn't vary that much. Where we see variation in color vision status and color vision types, that's usually in the genes that code for the medium and long wavelength opsin. And those are on the X chromosome. So I have a little schematic here of this X-linked opsin gene. And the amazing thing about this is that whether you have full trichromatic color vision or you're red-green colorblind, it largely depends on your sequence at just a few key parts of this gene, just a few key letters in the genetic code. And depending on what letters you have at these particular diagnostic sites of the X-linked opsin gene, that will largely determine what kind of color vision that you have. Now, part of what makes that interesting for me when I'm trying to study the color vision of wild animals, where I can't just give them a traditional color vision test, this means that you can determine the color vision status of an animal if you can get a DNA sample from them and just sequence them at the X-linked opsin gene. Basically, there's a simple genetic test for figuring out if an individual seems to have full trichromatic color vision or whether they seem to be red-green colorblind. So that's what we've been using in our research to assess color vision. Now, there's a long-standing debate about why is it that primates are the only mammals with trichromatic color vision, and why is color vision so variable across primates? And the adaptive value of having full trichromatic color vision, that's pretty obvious. If you're a primate foraging in a tree, it'll be much easier to spot ripe red fruit against a background of green forest foliage. And I think that's pretty well illustrated by this image here, where it's a doctored image, where on the left side, you see it as a trichromat, if you're trichromatic. And on the right side of the photo, you see that same tree as someone who is red-green colorblind would see it. And hopefully the difference is obvious, and that on the left side, it's easy to spot those red berries against the green background, but it's much more difficult on the left side, it's easier to spot those berries. The left side represents trichromatic vision. On the right side, which would represent how you'd see it if you're red-green colorblind, those berries are much more difficult to discern. But also, if you're a leaf-eating primate, it's still better to have full trichromatic color vision because you can identify new leaves, which tend to be red and more nutritious, compared to old, mature leaves, which tend to be green and less nutritious. So you can identify the most nutritious leaves.
Brenda, you're actually, we can't hear you. Let's see here. Oh. Oh, we will take a brief moment, and Brenda will be back. Brenda is going to be back momentarily. She is coming back onto the stream. If you have any questions already, please be sure to get them into the chat right now. It's going to be, there's so many different questions we can focus on, so be sure to get those in. And then as long as we're waiting, let's actually talk about our next episode of Lunch Break Science. We'll be featuring Shara Bailey on January 20th. She'll be talking about how we can learn so much about human evolution from teeth. It's going to be a really great episode. I'm going to go quickly check on things, and I will be back. Hi, Brenda. Welcome back.
I'm not sure what happened. My dog is sleeping, so I can't blame my dog for disconnecting the Ethernet cable. So,
we wouldn't be live if there weren't the potential of disaster. So, but we're glad to have you back. That's the important thing.
Yeah.
So why don't I give you a moment to get your slides back on?
All right. Do I need to re-upload them?
I think you can just hit share and slides, and you'll be able to. They should be already uploaded and ready for you.
All right, great. And can you tell me what the last thing you heard was? I'm not sure how long I went on.
Yeah, we're getting it queued up right now.
Okay, great.
Okay. Oops, sorry. Okay. I'll turn over the virtual floor back to you.
Okay, great. So hopefully it was clear as I was chatting about this slide that the advantage to having full trichromatic color vision, that's pretty obvious if you're foraging for red fruit against a background of green forest foliage. It would be easier to spot that red fruit against a green background, or even if you're a leaf eater, it would be easier to identify the new red, more nutritious leaves against a background of the more mature, less nutritious leaves. So whether you're a fruit eater or a leaf eater, there would be advantages to having full trichromatic color vision. But if full color vision is so great, why are so many primates and mammals red-green colorblind? And there's an idea that, well, perhaps there are adaptive advantages to being colorblind as well. And maybe those primates who are red-green colorblind are better at detecting camouflage prey or camouflage predators. And this idea comes initially from work done in human males, where they found that males who are red-green colorblind are often better at breaking certain types of camouflage. And there's some empirical evidence for this in non-human primates as well through work done on cebus monkeys by Amanda Mellon and her group. So to kind of illustrate this potential value in being red-green colorblind, here you see this field of flowers and the red tulips immediately draw your attention. But if you look at this field as someone who's red-green colorblind would see it, the little purpley blue flowers that are also in the field become much more apparent. Okay, so as I said, color vision is variable across the primate order, but humans, apes, and the monkeys of Asia and Africa, all, for the most part, all generally have routine trichromatic full color vision the way that we do. In the monkeys of Central and South America, their color vision is described as being polymorphic. So virtually all of the males are red-green colorblind, and then, but then some of the females have trichromatic color vision. So this is described as being polymorphic, and it's thought to be a similar scenario for lemurs, lorises, and galagos. So that's what we were really interested in, is trying to understand the variation of aspects of color vision in lemurs. So we've done a survey of color vision using these genetic diagnostic tests. So the survey has included 18 species at 31 different sites from across Madagascar, and we've found that color vision is, in lemurs, just highly variable, not only across species and across sites, but also within species. So here I'm going to give you an example for one particular group of these lemurs. These are Shafaka lemurs. So what you see here is a little map of the different sites of Shafaka lemurs where we've characterized their color vision. So we have 11 different populations, 11 different sites where we've characterized their color vision. And where you see an icon, like the little rainbow icon, that means that within that population, they have the variant that would allow for full trichromatic color vision. So most of these Shafaka lemurs, most of these populations indeed have that variant that allows for full trichromatic color vision. But notably, you can see these three populations in the south, they lack that variant. So all of the lemurs in those three populations, all of the lemurs are red-green colorblind. And the interesting thing here is that these three populations in the south are the same species as two that are farther north. So for a single species, we have two populations where they have the full color vision variant, and then three populations in which all of the individuals are red-green colorblind. So it's, again, variation not just across species, but even within species, variation across populations. So when we've mapped this out for all of our lemurs and tested, well, what are the ecological factors that might predict this variation in color vision or be associated with this variation in color vision, there are two things that fall out as being highly predictive. One is activity cycle. That's not surprising. You would expect that with nocturnal lemurs, they're going to be far less likely to have full trichromatic color vision. The other thing that falls out is habitat as being a predictor of variation in color vision. And this also makes sense if you consider the habitat types of Madagascar. So Madagascar has these kind of three primary habitat types. You have evergreen forest in the east, deciduous forest in the west, and then in the southwest, you have this unique spiny forest habitat. So now I'm going to add, I showed you these results for the Shafaka lemurs a minute ago. So now I'm going to make the point on here that you'll remember with Roy Shafak, with this one particular species, there were two populations that had the capacity for full color vision. Well, those are two populations living in deciduous forest. These three populations where everyone's red-green colorblind, well, those are all populations in the spiny forest. And we see something similar for ring-tailed lemurs. The population in the deciduous forest has the capacity for full color vision. The population in the spiny forest, everyone's red-green colorblind. So these are different species, but you see a similar pattern based on habitat. So it seems that differences in habitat are perhaps helping to drive differences in color vision across populations and across species. And I just wanted to wrap this up by showing a picture of this spiny forest in Madagascar. And it is something, I'm not sure this picture does it justice, because it is a place that looks like it was designed by Dr. Seuss. It is amazing. And I wanted to end on this note by, again, making the point that although in our lab we might be studying these genes, these opsin genes, for us to ultimately understand the importance of the genetic variation that we're seeing in the lab, we have to understand the animals in their natural environment. And what's really crucial to our research are our collaborations with field primatologists who are studying the animals in the wild and providing that connection. So on that note, I would like to really thank all of those many collaborators. And I especially want to thank Jason Kamilar, Nick Mundy, and Rachel Jacobs, who have been key parts, particularly of the Color Vision project. And yeah, of course, I thank all of you for listening and for staying on through my technical difficulties.
Thank you so much, Brenda. That was a wonderful talk. Before we take questions from the audience, we're actually going to give our viewers the opportunity to test their color perception. So Brenda will share an image. You will all of you viewers out there will carefully observe the characteristics of the image and submit your assessment of what pattern you see in this image. Okay, so let's get this our first round. So yeah, what is this?
So most of you have probably seen something like this before. These are called Ishihara plates. They are a traditional means of testing for color vision capabilities.
Okay, let's see. Do we have any assessments yet? Okay. We have one. Let's see here. We have two. We have another one. Let's see here. That it seems like they are all 45. Okay, let's see what the correct.
Yes, 45. And if you have typical routine full color vision, routine trichromacy, then yeah, you should see a 45. I saw a 45. Ariel, did you see a 45?
I did see a 45, yes. So now we have a second round.
It's another Ishihara plate, but one that people might be less familiar with.
So like the first round, just note what pattern you see in this plate. It's called a plate, correct?
Yes.
Okay. That's very different than the previous. We're not seeing any responses yet. I think this one's a little stumping for some of our viewers. Let's see here.
It is true that it will be a very small fraction of the population that will be able to answer this.
Okay, we have one comment so far. Let's see here. No idea. No idea. Let's see here. We got some more. Oh. No, don't know what to name it. What's wrong with my cones?
Yeah, so if you were able to see the 45 in the first plate, then you probably won't be able to see anything in this plate. So whereas the first plate was testing, do you have trichromatic vision? If you could see the 45, that's an indication that you do. This second plate should indicate if you have red-green color blindness, you should be able to see a 5 in this Ishihara plate that those of us with routine trichromacy can't see. Again, it's going to be a small fraction of the population that is able to see this, but it's worth noting that we're all familiar with the Ishihara plates with the numbers and the letters that are easy for us to do, but those of us with trichromacy to do. But there are also the flip side of that. There are plates where people who are red-green colorblind can discern something that we can't. So I think it's important to keep in mind that both types of color vision can have their advantages.
Well, it seems like the vast majority of our viewers, at least the ones that had submitted assessments, are not red-green colorblind. But
Yeah.
if you are watching this video on the replay, please be sure to drop a note in the comments if you were able to see the pattern in that last plate. Okay, so let's get on to our audience questions. If you have not submitted your question for Brenda yet, please be sure to do so right now, because it's now or never with questions. Okay, so I know we have a couple of questions here. So or who is our first question come from? Again, okay, from Pete. Okay, it's a long question, but it is essentially, are there genetic codes associated with night vision? Because that would obviously be a wonderful benefit for seeing at night.
Yeah, so there are a couple parts to this question. So our night vision, this discriminating between light, that's relies much more on the rods than the cones. And the fact that our cones, our ability to discriminate colors with our cones isn't very useful at night is anybody who's had, where I used to live, I had to take out my recycling and trash at night, and I had a green bin and a blue bin. But if it was a dark night and I went out there, I couldn't tell which was which. I had to bring a flashlight to figure out which was blue and which was green. So in general, our cones aren't functioning that much at night. And that is probably part of the reason why nocturnal primates, you don't see this trichromatic color vision, because it just really wouldn't be that useful for them. And then there was a second part of the question about Neanderthals. And I'm fairly certain, although I don't know this specifically, but I'm fairly certain that Neanderthals have the same variants of the, what we would consider the quote unquote normal variants of the X-linked opsin genes that we do. So I think it's safe to assume that they had trichromatic color vision just as we do.
Okay, do we have our next question? Okay. Our next question is from Life Out in the Farms, and it is, what's the role of stomach in a human evolution?
Yeah, so that's interesting. Stomach is, we do projects on almost every phenotypic realm in my lab, but not much on the stomach. We have a few students who've done a bit in looking at the microbiome or digestive enzymes, the evolution of genes that code for digestive enzymes. So yeah, so there are a lot of levels at which to answer that. So, you know, the stomach is playing a key role in breaking down what you're ingesting. And we have this old school idea that it's our cells, our genes within the stomach that are doing that. But we've become much more appreciative of the fact that it's also our microbiome, all of those enzymes and proteins that are created by the bacteria in our gut that are playing a big role in breaking down the stomach. So any question about the evolution of the stomach also has to include a conversation about the evolution of the microbiome. And that's something that's relatively new. That's not something we were talking about much, 5-10 years ago.
Well, so it sounds like a topic for a whole nother episode.
Yeah.
So we'll duly note that question. Let's see here. Our next question comes from Camilla. Is there a theory about why the lemurs in the Spiny Forest have different vision?
Yeah, so the structure of that forest is completely different. So all I can say now is that we have this observation that in the Spiny Forest, everyone's red-green colorblind. And so our best guess is that that ancestral population had the polymorphism because other populations of the same species did. But for some reason, it's lost. Maybe it's lost because the selective pressure to maintain that full trichromatic color vision just isn't useful in that environment. It could be that it was lost, that the variants that allow for full trichromatic color vision, that they were just lost through neutral forces like genetic drift. But if you look at other regions of the genome of these lemurs, there's not an indication that they went through that type of bottleneck or that they have less genetic diversity compared to the other populations. So I can't give a specific answer that it's XYZ aspect of that habitat that would have led to a reduction in either pressure for maintaining the trichromacy or direct selection for being red-green colorblind. But one thing we really hope to do, and again, in collaboration with the field primatologists, are for those populations where they have the variation, to fully characterize, well, which individuals have full trichromatic color vision and which individuals have the various different types of red-green color blindness, and then use the long-term data to see how that impacts aspects of reproductive success and behavioral movements and who eats where and who leads group movements. That makes sense. So hopefully by combining the genotype scores with long-term behavioral analysis, we'll be able to get at, okay, what might be the value, either the value or the lack of, in having these different types of color vision.
Interesting. You've talked a lot about collaboration. What is the importance of collaboration, and how do you establish collaborations?
I mean, for our lab, they're everything. Really, the most important piece of equipment in our lab are our -80 freezers. And we have DNA from, I don't know, 900 and some animals, representing 96 species, I think. And all of that has come about through collaborations with primatologists working in the field, working in captivity, with collaborations at zoos. So yeah, that is our most precious resource. But then all of our biobank then is still shared with the people that collected those samples, because the samples on their own aren't that interesting. It's only if you have that matched phenotypic data, the long-term data about what the animal looked like, how it behaved, aspects of its ecology, that then you can link back to the genetic variation that makes the samples worthwhile. And so that's the data being generated by the field primatologist. So again, yeah, all of our projects, although I'm largely based in the lab, the key to most of our projects is what's happening in the field.
Oh, it's just got to be so interesting to get to work with so many different, and do people come to you, or do you like reach out to projects, or do you get new students, and I don't know.
Yeah, it's a bit of both. And yeah, I've been very fortunate in having great students who themselves will, foster new connections. And in fact, that's been a big benefit of going to conferences for us, because we'll hear someone give a talk and we'll be like, oh, I wonder if they would want to collaborate on XYZ, because look at what they're studying. If we could sequence their whatever serotonin receptors, that might be informative for them. So in fact, I would guess it's conferences are where we tend to foster these collaborations, where we tend to identify links we might not have seen before.
Oh, it's got to be so fun. And we have a question from Cheryl. You mentioned the benefits of vision for foraging. What about for mating? And also, do lemur infants have different fur colors from their parents?
Yeah, so for that first question, yeah, almost certainly. Well, let me take a step back. I mentioned early on that part of what I find fascinating about primates is their variation in color. And the fact that primates are highly variable in their color and are the only mammals that have trichromatic color vision, those are not a coincidence, I don't think. I think the evolution of trichromatic color vision probably came about more through foraging and things like that. But once it's there, then it could be co-opted for all of these aspects of sexual signaling, like large sexual swellings or red hair ornaments. So I think that these things are definitely connected. And that's why these two seemingly different lines of research in the lab are often intersecting. And then what was the second part of that? So almost certainly they're playing a role in mating. And that's something that whenever we're analyzing, if we're studying hair patterns and we're trying to ask, is this something that could have evolved through sexual selection? Then we're often curious to know, okay, well, what is the genetic basis of that, and how, with the different types of color vision of these conspecifics, how would they view these phenotypic types differently? Because how they, if you think about it, how monkeys are viewing each other, how they see each other, if they don't have, in cases where they don't, or lemurs, where they don't have trichromacy, they view each other differently than how we see them with our trichromatic vision. So.
Really interesting. My last question for you is, do you have any advice for those who are just starting out or hoping to pursue a career in science?
Yeah, I've thought about what of the non-obvious things that have promoted real moments of fun or productivity in my career. I realize so many of those have come about through having conversations about my research with non-experts. So, we're always talking about, it's those conversations with non-experts that often lead to new ideas or really thought-provoking, um, the thought-provoking conversations that lead me to maybe think about my research in a way that I hadn't before. We're always talking about our research with other biological anthropologists, but that can almost become a little bit of an echo chamber. But when you're talking about your research to interested non-experts, that's often where major breakthroughs can happen. So that would be my advice. I would say really hop on any opportunity you get to discuss or present your research to non-expert audiences. Because that can be a gold mine of new ideas. And, the Leakey Foundation is so great at providing exactly those opportunities. I sit on the SEC and I get to go to a lot of Leakey events, and I always come away from them feeling that those conversations were really stimulating and thought-provoking. And so I'm grateful to the Leakey Foundation for that, and for hosting things like Lunch Break Science.
Well, thank you so much. And Brenda, this was just a phenomenal episode. I'm so glad you could join us for our first episode of 2022.
Thank you so much for having me.
So next time on Lunch Break Science, we will be featuring Dr. Shara Bailey, who will be discussing teeth and what we can learn about them about human evolution. So be sure to check in for that episode. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge.
Lunch Break Science is brought to you by the Leakey Foundation and is made possible by the generous support of the Ann and Gordon Getty Foundation and Camilla and George Smith. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientist, and how you can help support human evolution research and educational programs like Lunch Break Science. Right now, all donations will be matched by generous donors, meaning your impact will be doubled. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming episodes and programs, as well as groundbreaking discoveries in human evolution research.